So I want to thank the organizers of this conference, especially Deborah Kahn, for the invitation to speak with you today. Title is Ethics of Scientific Publication, Deficient Institutional Responses. And those institutions are both the institutions where the research is conducted, but also the institutions of publications. So I've been interested in the sociology of science for a very long time, but my recent incarnation as a scientific detective starts here. This is Mono Lake in California. And there was an article in Science in December 2010, which is referred to as the arsenic bacteria fiasco. And because I worked on a related field, I knew immediately that this was a hoax, and that, and I started working on this, and I was able to identify the fact that at every single level, peer review, the institutions, the media, there was failure, absolute and abysmal failure to deal with this issue. So I would go around internationally and speak about what was wrong. What did this tell us about peer review? What did this tell us about the relationship between science and the media? And people would come up to me afterwards and say, "You think that's bad? You should take a look at this." And one of the examples that they gave me was on this next slide here, a PNAS article. And I'm a college professor. I believe in audience participation. So I'd like you to take a look at this image and see if you can tell me what's wrong with it. Yes, back there. Shout out.
But the first and most important is that they're clipped. You don't see the full western block.
So that's true. This is just a, I mean, this would be okay, for example, if in the supplementary material, they gave you the whole blot, right? That would make it okay. This was before the time that was common, but that's not what I'm focusing on per se. Because again, this was fairly common at its time. I'd like to draw your attention to panel B, specifically to the beta-actin, the one that's labeled beta-actin. Does anyone see anything there? Go ahead, shout out. Yes, yes. So 3, 4, and 5 have been duplicated in 6, 7, and 8. All right? This cannot happen by accident. Okay, this isn't just a mislabeling issue. This has to be misconduct. It has to be true image manipulation. So I contacted the journal about this. They went to 2 experts, one of whom said, yeah, there's clearly a duplication, and the other one says, I don't think there's a duplication. So they decided just to drop it. And the authors denied that there was any issue here. I started thinking about this and the fact that there was a denial, and so I started looking into articles by the same group. And I was able to identify about 30 articles that had either image manipulation and or plagiarism. I'll just mention that if you draw a Venn diagram of those papers from labs that have image manipulation and those that have plagiarism, there's a fair amount of overlap in that Venn diagram. But in any case, 30 articles in which there were problematic images and or plagiarism. And I do all of this manually. I don't use any computer program to identify this. I just look at images. And I don't know, there's something wrong with me. There's a part of my brain that seems to store these images and to compare them. And when I was recently at the Wallace Collection in London, I was looking at the wall, and then there was a painting by Titian of Perseus and Andromeda on the left, and then there's another painting by François Lemoine, which is about 150 years later, also of Perseus and Andromeda. Now, it's clear that they came from a common origin. All right, you've got the same, I mean, there are mirror images, you've got the castle in the background, you've got the strange-looking water monster in the middle, she's tied up. I can't tell you that Lemoine, saw the Titian. He probably, if he did see it, he probably saw it in a print form, or that they both came from a common ancestor, that there was something. But there's no question that these two images are related. They have a common origin. And I'm pretty sure that the Wallace Collection collected them precisely because of that. All right. I'll have more to say about this in a little while. This is another article from the same group. And I'll ask once again, does anybody see anything puzzling about this, this image? And I'll focus you on E and F, panels E and F. Anyone? I'll focus you on RUN6. This is a northern blot. Right, so RNU6, the one in E, is the same as the panel on the right-hand side, the RNU6 of panel F. Does everyone see that? So, as I said, we're talking about 30 articles from this group. I wrote to the journals about every single one of these articles. And in most cases, I got little or no response. I mean, they'd respond initially and then say, oh, there's nothing there, we're not going to do anything, so on and so forth. The authors say that there isn't any problem. In this case, this is JEM, it's incredible. Within a month, there was an online correction of this article, within a month of my sending the letter. And that's shown here. So, there we go. You can see that this and this do not correspond anymore. So they're different panels. But there's still a problem. And so I wrote back to the journal and said, congratulations, this is incredibly rapid response. I really appreciate it. I still have a little problem. My first problem I have is that the RNU6 panel is the same as it was before, except it's inverted. And I'll show you that in a moment. How do you know it's right now and it wasn't right before? They said, case is closed. We know. So I said, okay, I have another problem. Says, remember I told you about that part of my brain that stores images? I had dealt with this article from Cell Press previously, and I had actually achieved some corrections for it. Not all the corrections that were necessary, but achieved some corrections. And so it was already present in my mind. And I remembered this image over here. And by the way, oh, I'm sorry. There are bands here, but we just can't see them. I can see them on my screen here, but we can't see them here because of the display. I'll have something to say about that in just a moment. But just to show you first, this is the first panel, and this is the correction. As you can see, that it's just flipped. I want to refer to these things as, I hope this is okay, submarines with periscopes. And so you can see, for example, this one, the periscope is a little right of center. Here it's a little left of center. These are mirror images. Here is the corrected panel, all right? And here is a flip of the one from a previous paper 3 years earlier with a subset of common authors, including the first and last authors. There's no question that this is a duplication. Again, I could run you through the periscopes, but it's obvious. So I wrote back to JEM, I also wrote to this journal as well, and said, I don't think you should close the case. There's a real problem here. And the problem here is that they know, the authors know, that there's some terrible person out there looking at their images, okay? And I will share with you, they not only know that there's some person, but some of the editors-in-chiefs violated my privacy and identified me to the authors. So they know who I am exactly. And Yet, they had, I'm sorry, I'm going to use a technical term, the chutzpah, to submit a correcting image that is taken from a previous publication. So I wrote back to them, and they said, well, we'll take a look at this. Here's another example. So because I wasn't getting action from the journals, I used contacts at the New York Times to bring this to public attention. And there was a front page article in the New York Times in March of 2017 about my efforts in this laboratory. And so the principal of that article with an AL, principal was decided to sue me, decided to sue the New York Times, decided to sue his own institution. He lost all of those suits at summary judgment. But discovery is a wonderful thing. And in the process of discovery, I learned all sorts of interesting things. So here's another one. This is a heat map expression. Does anybody see anything? Focus on B for the moment. Does anyone see anything interesting about panel B? And so I'd encourage you to compare the two parts of B, the left-hand red and green stuff with the right-hand red and green stuff. Does anybody see anything there? Right? It's just transposed up. That can't happen. Okay? It cannot be. I mean, if I did this experiment on 2 different days, I wouldn't be able to get the exact same numbers out of it. So it's been transposed. This is figure number 1. The entire paper is based upon these data. That's what's shown in panel C is based on B, and everything else in the paper is based on this, a Nature Medicine paper. This just another example. Any data can be faked, okay? It's not. That's just part of what I'm trying to display here. But it took. So there were lawsuits. I also wound up reporting these papers to the institution, and after a fairly long process, what they wound up is finding the 2 postdocs who were the first authors on this paper and the one I showed you before, guilty of misconduct. They did not find the PI guilty of misconduct. I will tell you, again, the vote was close, and especially this issue about submitting bad data the second time, all right? It's okay, one can understand perhaps that you aren't always paying attention to all the data when it's first submitted. I mean, this one's obvious. I mean, and the problem is, how many people are on this paper? I don't know, 12 people on this paper? None of them apparently looked at the figure, right? It takes 2 seconds to see that there's something wrong with this figure. But once it's pointed out to you, and you and just say that postdoc are responsible for the correction, and you don't do the due diligence to make sure that you're not submitting data that you had previously incorporated in another paper, it seems problematic. Okay, so that's the end of that story for now. This is another example, and it's not so clear again on this image. We're missing part of it, but there was a series of papers from an individual who's now at the Center for Genomic Regulation in Barcelona. And I've given this talk a number of times. You can see, in fact, that these boxed regions are the same band. There's actually additional features. Again, to talk technically, it's not necessarily the band. Bands look pretty much the same. It's the schmutz around the bands that tell you that there's a problem. So as you can see, this is 20, 45, and 50. What the Center for Genomic Regulation in Barcelona did was to get an outside computer image analyst to see whether this, in fact, was the same image. And so the analyst did this, and what you can see is they've got 50 and 45. I mean, pixel by pixel, it's identical. Right? Pixel by pixel, it's identical. Whereas this one down here, 25 looks completely distinct. But the problem with that is, it should be band 20, not 25, that he was supposed to analyze. And neither he, nor the institution, nor the journal seem to have noticed this when they decided to dismiss this case. So this is pixel by pixel identical, even 50 and 45. But his assignment was not to determine whether these are the same. His assignment was to find any evidence that they were not the same. So here's the next image. Look, there's something called data compression, and as you copy data, things happen. Here is you can't get anything that will be closer, You've got Crete down here with a little sun over it. These are identical images. And the fact that there were some random arrows pointing to some sort of difference makes no difference. The question is not, can I find some trivial difference between images and therefore say they don't have a common origin? The question is, how likely is it that two images are going to be so similar and yet not have a common origin, right? If I have two, photos of the Mona Lisa, one can be pretty confident that it is either a photo of the Mona Lisa or a photo of the Mona Lisa or some other representation of the Mona Lisa. It doesn't matter that I can find some sort of small difference because of the reflection on the glass or whatever it is. Okay. Unable to get any action on this. And this, again, one of the things that concerns me, I get all sorts of people contacting me all the time. And I'm not, people can make mistakes, right? I can get mistakes. None of these are possibly mistakes. Okay, there is no possibility of honest error. And I don't tend to concern myself with ones that could possibly be honest error. And I also tend to concern myself only with ones where there are multiple instances from a group of problematic data or plagiarism. Okay, this is a pretty recent case. So these are microscopy images, and just in brief, these are two different cell lines, okay? Two different cell lines where an experiment has been conducted. And I've made this easy. I've boxed. You can see that this here is the same as this here. This here is the same as this here. These little boxes here are blown up over here. You can see this ice cream cone, partially bent ice cream cone, is the same one as here, and the little part has fallen off. These are the same images. That's not possible. Okay, it's not possible. And furthermore, what happens is this image here is a different field. Okay, so on the bottom panel, the one on the left, the next one are the same as on top, but the third one is from a different image, a different field. So when they do the merge, of course there isn't going to be any overlap because they're looking at different cells. Okay, but they make a conclusion on the basis of that. And some more. This is Sri Lanka with a couple of lined, things coming off, and you can see that this and this are the same as well. Contact the journal. By the way, this isn't the only problem with this image. This is a American Society for Microbiology journal. And they say, I get back, we contacted the authors, they explained what happened, no problem. Well, there's no possible explanation of this, okay, that you cannot explain this. And even if it were a correction, an honest error, it still needs to be corrected. No correction whatsoever. No acknowledgement that there was even a problem. All right, I'm going to finish. This is going to be my last example. These are two tables from two articles in the same journal. And I want on my gravestone to be inscribed, discoverer of ovarian cancer in males. Now you're probably saying to yourself, men don't have ovaries, and you're probably maybe you're thinking, well, are we talking about trans? No, we're not talking about that. What you can see on this one on the right, this is microRNAs. Oh, by the way, in case you don't know, microRNAs in cancer is one of the fields which is most dominated by paper mill products. We're talking about thousands, if not 10,000 articles. I mean, it's ridiculous. Okay, so what you can see in this table is that 19 of the patients who have ovarian cancer are male. I have another article that also says, I'm going to have to say, I have female prostate cancer as well. I have articles with that. So using just sort of techniques, I was able to quickly find that there's another article from the same journal about thyroid cancer with the exact same panel in it. As it happens, also a paper mill product. There are problematic figures in this as well. Contact the journal. Journal says thank you, puts a little note, we are investigating this. What's to investigate? 30 seconds should lead you to a retraction. And this particular journal has very problematic peer review. By problematic, I mean next to non-existent. And 2 years later, the article had still not been, neither article had been retracted. Just had this little note. Continue to be cited. Articles that get retracted also get cited, but that's another issue. No action. I eventually decide, you know how I used the media last time? Well, now I decided to use an undergraduate student. It's interesting, the responses I get. Sometimes, because people know who I am when I'm writing, I get a good response. And sometimes, because they know who I am when I'm writing, they just try to blow me off. And they, oh my God, it's him again. And so, I'm just by the way, a number of us have different types of styles. When I write, I'm very polite, and I never accuse any individual. I just say, hey, what action are you going to take about this? That's how I finish my emails. But this undergraduate wrote a note to the editor-in-chief and said, I'm an undergrad, I'm interested in science, I believe in science, but I ran into these articles. How can you have an article in your journal that says there's ovarian cancer in males? And it's identical to this. No action. She then decided to write to all the members of the editorial board and ask them two simple questions. Question number one is, did you know you were on the editorial board of this journal? And the second question is, do you realize what sort of thing is being published in your journal? And many of them said, no, I didn't realize. I've never reviewed. This was a while ago, and I may have agreed to it, so on and so forth. And she was keeping snapshots of the editorial board over time, and you could see people dropping off because of action. So they decided finally to retract these two papers. So you have to use these different techniques. So I want to point out just two things. How much more time do I have? 10 minutes, great. I am not anti-science. There are some people who work in my field, in this field about research integrity, that have a chip on their shoulders about science or so on and so on. I'm not trying to discredit science. I am a scientist. It happens I'm also a West Lafayette City Councilor. I use science in my approach to government affairs. I'm a huge advocate for vaccination. I work on Ebola virus and, as you heard, virus-based gene transfer, gene therapy. So I'm not anti-science. I believe naively that science is like a higher endeavor. And if it is a higher endeavor, we need to adhere to higher standards. It may be that the rest of the world out there no longer believes in the idea of truth. I believe that scientists need to adhere to that. And that's why I'm in this. And I believe we can. So let's talk about some solutions to these problems. And I write for something called Times Higher Education. It's the European equivalent of the Chronicle of Higher Education. You can find a number of my articles there, but I've been reading, I've been talking and writing about these issues for a long time. So let's talk about peer review. And to paraphrase my graduate advisor, who was Dan Koshland, who was the editor of Science for 10 years, by the way, he would not have accepted that arsenic bacteria paper, and it also has not been retracted. But that's an aside. I'm paraphrasing him, who paraphrased Winston Churchill, which is that peer review is the worst possible means for determining whether an article should be accepted for publication, except for all the other ones that have been tried heretofore. So, which I agree with it. But there were lots of problems with peer review. So I mentioned, I spoke out yesterday, as I speak out about things, that I think we do peer review exactly backwards. And that is, I believe that articles should be written in the third person, that when an article is sent out for review, the names of the authors and the institutions that they're associated with should not be included with the article. There is incredible bias that's introduced into peer review by having the authors and the institutions they're associated with known to the authors. Now, I'm not saying it's irrelevant, right? After you do the review, you should then get the names, because that may influence you. For example, you'll say, well, now I know why they cite this paper that nobody else in the field believes. Or now I understand how this fits in. So I can understand, I'm not saying that the information is irrelevant. I just think it shouldn't be determinative on whether you decide to accept the review and influence you when you do the review. Contrary-wise, as I mentioned yesterday, I believe that peer reviews should be published in the online version of the article with the names of the reviewers. And just to talk a little bit more about yesterday, what is the reward for doing a good job at peer review? Zilch. Okay, there is no reward. What's the punishment if you do a bad job at peer review? The worst thing that will happen to you is that particular editor won't ask you to do it again. And for most people, that's not a punishment. Okay? So, as I mentioned before, I have this scheme that we include peer review as part of our contribution to the scientific endeavor on our CVs, and it should be included when we are being judged. Which brings me to post-publication review. There is nothing privileged about pre-publication review. It is not necessarily better quality. It is, just because an article is published doesn't mean that peer review is over. And post-publication review can be at least as good quality, and it should be immediately responded to. And I often get, I'll just be honest here, no editor or editor-in-chief has ever decided that they wanted to occupy that position in order to deal with bad research that has previously been published. That's simply not their interest. They want, there's a prestige element, but there's also, you get to be involved with people, you get to see research before it becomes public, all sorts of advantages. And you can, as I learned through the discovery process, you can trade favors with editors of other journals. But this is part of your responsibility. And all the time I hear, we don't have enough resources to deal with all these allegations and so on. And it's not our role to determine whether there has been, image manipulation or image duplication or so on and so forth. My argument is, if you don't have the resources to clean up the literature that you've already published, you don't have the resources to publish new information, because you're simply not taking a responsible approach to it. This has to be part of the job. And post-publication review is an extremely important part of that. Now, in 1994, when I was at the Whitehead Institute, I gave a seminar on how to transform scientific publications. I don't know how many of you remember FTP and Gopher, but this was just before the World Wide Web, 1994, just around the time. So I had this really cumbersome system of using FTP in order to have articles, and you receive an email telling you that these are the articles that were coming out. But I also had a comment section. This was going to be embedded in this, that people, it was going to be curated, but the idea would be that there would be allowed comments, post-publication comments, for every article, because I believe in the literature as a dynamic entity, rather than, oh, I got it published, it's untouchable now. All right, so post-publication review, just as important as pre-publication review. Anonymity. I actually don't believe in anonymity. Okay? I understand, having been sued, why one might want to do that. And I understand people are concerned, I'm junior, whatever. This is all of our responsibility. This is the responsibility of everyone in science. When you see something, say something. Okay? And say it under your name, because transparency is important. If I'm going to say that there are 30 articles from a particular lab that have problems, I should be willing to stand by that, as a scientist. So I don't believe in anonymity of peer review. I have friends and colleagues who do this sort of thing anonymously. I don't think in the long run that's actually to the advantage of science. Conflicts of interest. Everyone in this process has conflicts of interest. Okay? The journals, they don't want to devote the resources. They don't want to look bad by having their retraction numbers up. They don't want to alienate authors. Okay? Some of these authors are prominent authors. They don't want to alienate them, and so therefore they won't pursue these things. And so research institutions have a tremendous conflict of interest, right? They have money on the line. Okay, if a person is found guilty of misconduct, they may have to pay back federal funds. So conflicts of interest, we have to eliminate them. We have to eliminate them. So what are the sorts of things that are necessary? When we're talking about research integrity for journals, the person needs to be semi-independent. I know that somebody has to pay his or her salary, but they have to be focused on the science and not the interests of the journal. When I've dealt with some of these, they vary. Some of them really are like that. And they're given the independence to pursue things to the end, and they take a responsibility. Other ones, research integrity seems to be a part-time job. They have other ethics parts of their portfolio as if that was one thing. And so they need semi-independence. The same thing is true with institutions. You may be familiar that institutions have a two-phase process for investigating these allegations. They're called inquiry and investigation. Inquiry is just to determine, is there anything that's worthy of an investigation, and then they have the investigation. And I can understand one gets all sorts of allegations, and you might want to do the inquiry in-house just to sort through that. But the investigation should be done by outside investigators, not investigators at the institution. There is just too much defending of the institution by people who are inside. So there can't be a true, thorough investigation. Finally, what's the new approach? And the new approach is everybody does this all the time. This is not for me to do. I don't need to do this. I have other things to do in my life. But people bring things to me, and they're junior, they're afraid, they don't have the expertise to look at these things. we all have to be involved. And we also all have to stop believing that the computational tools that we use are exclusive. So that is, if something is found to be negative using some computational tool, for example, plagiarism detection, it doesn't mean that it's actually not plagiarized. Okay, you have to go beyond those tools. You actually have to understand how those tools, the limitations of those tools, and how to use them. So we all have to be involved in this. It's not just the journals. Every single scientist has to see this as part of their responsibility. Finally, this is also by François Lemoine, and it's called Time Saving Truth from Falsehood and Envy. And those articles that I mentioned, so the one with the submarines, the one that the 2010 paper has now been retracted. The other one only has an expression of concern. The one with the heat map has now been retracted, but we're talking about 6, 7 years. So time saves truth from falsehood and envy. So I feel positively you have to stick with it and that you can get change over time. The problem is, François Lemoine, this was his last painting. He committed suicide immediately after this painting. So perhaps, even though he painted this, he wasn't fully confident that this was in fact the case. And so I'm not going away, but I really think that you need to be working more with those of us who are concerned about these issues. And I appreciate your patient listening to my critique of the publication ecosystem. Thank you very much.